of our cells which are involved in the presentation of the antigen. Antigen is actually coming from the pathogen. So how they present the antigen, we are going to discuss that. Then finally, we'll end up discussing how the T cell activation occurs and what are the effective functions of the T cell by which we are able to get rid of the infectious disease. Similarly, another arm of the acquired immune system, B cell activation and its effective functions. So this is what we're going to cover in this session. Now, we all know that almost all immune cells in a body arise from the pluripotent stem cells. And these pluripotent stem cells are arising in the bone marrow. So practically all immune cells arise from the bone marrow, and there are different lineages which are there. So we've got a lymphoid lineage, as the name suggests, lymphoid lineage. Lymphocytes arise from these progenitor cells. Then we've got myeloid lineage, from which phagocytes and phagocytes, which includes both monocytes, macrophages, and granulocytes. Granulocytes, we all know, it includes eosinophils, basophils, and neutrophils. Okay. Now, if you talk about lymphocytes, we've been discussing it earlier also. Lymphocytes are further subdivided into cells such as T cells, B cells, and NK cells. Remember, we last time discussed about NK cells, that is the natural killer cells. And then phagocytes, as I had mentioned earlier, monocytes, macrophages, and then we've got granulocytes. Then, in addition to that, we've got accessory cells. These are referred to as antigen-presenting cells. The origin of these cells actually is a bit of an enigma. We really don't know from where they arise, but people think that they also arise from bone marrow. And the most important accessory cell that we need to remember is an antigen-presenting cell. As the name says, the cell is involved in the presentation of the antigen. Thereafter, it kicks off the immune response. So we're going to discuss all this in subsequent session. So this just shows the origin of various immune cells. So this blue thing is the bone marrow depicted, and then we've got a pluripotent hematopoietic stem cell. Then it further divides into a myeloid progenitor or a lymphoid. Lymphoid, as we've discussed, what are the cells which arise from lymphoid lineage, and then myeloid lineage. Please remember, myeloid lineage not only gives rise to the monocytes, macrophages, and granulocytes, it also gives rise to your platelets as well as the erythrocytes. Okay, all these things would also be covered in your pathology classes. Okay, when we talk about hematology. If you talk about in detail about T lymphocytes, because T lymphocytes are part of the acquired immune system, they have to recognize a specific antigen. Then only they will be able to develop an immune response. Remember, adaptive or acquired immune response has to specifically recognize the antigen. And the surface molecule which helps it to do so is a TCR, which is in short referred to as T cell receptor. TCR is very intimately associated with another molecule which is called as a CD3 molecule. CD, we know, stands for cluster of differentiation. Okay, and T cells have further divided into two subpopulations, that is Cd4 positive T cells, which are also referred to as helper T cells, and CD8 positive T cells. If you talk about Cd4 positive T cells, which are also referred to as helper T cells, they form roughly 2/3 of all the circulating T cells in the body are helper T cells. The T cell receptor, which is expressed on the surface of the helper T cell, recognizes the peptide antigen, which is presented by self-MHC class 2. We'll talk about MHC as we move along. So this is a rule in immunology that you have to remember. CD4 positive T cell cannot recognize a free antigen circulating. It can only recognize an antigen which is processed and then presented by our own self MHC class 2. So this is what we refer to as MHC restriction. So if I say that T helper cell is restricted to which MHC, can you give an answer? Quick, if you are listening. CD4 positive T cell or helper T cell is restricted to which MHC? You will say it is restricted to MHC class 2. It means that it can only recognize antigen which has been processed and then presented in the context of self MHC class 2. Okay. Once the helper T cell gets activated, It further subdivides into two functional subsets, Th1 and Th2. Th1 cells will see activate the macrophages and they bring about what we refer to as cell-mediated immune response. On the other hand, Th2 cells activate B lymphocytes. And these B lymphocytes, once they receive help from Th2 cell, they then differentiate into plasma cells. And as we discussed last time also, and your earlier sessions also, plasma cells then secrete the antibodies, which are also referred to as immunoglobulins. And these and then are secreted into the circulation, and they are able to bind the free antigen. Okay, so the antibodies are able to bind the free antigen or cell-bound antigen, but that antigen need not be processed. Okay, unlike what we referred to with regard to T helper cell, it can only recognize antigen which is presented in context with MHC class 2. Let's move our attention to CD8 positive T cell. CD8 positive T cell is also referred to as cytotoxic T cell, and we'll try to understand the meaning of this word cytotoxic. It's pretty easy. Cyto means cell, toxic means killing it or damaging it. So CD8 positive T cell directly recognizes the antigen which is presented by a cell, which we'll discuss in a short while, and then kills that cell, whichever cell is presenting the antigen to it. So that's why in that context it is called as cytotoxic. Cyto means cell, toxic means damaging it. 1/3 of the circulating T lymphocytes are CD8 positive T cells. In contrast to what we discussed with regard to helper T cells, CD8 positive T cells, they recognize antigen in context with the MHC class 1. So similar to what we discussed for helper T cells, it cannot recognize antigen which is freely circulating. It has to recognize an antigen only when it is processed and presented by self-MHC class 1. Hence, if I ask this question, CD8 positive T cells are restricted to which MHC molecule? Quick answer. Can I get a quick answer from you? Very good. So they are restricted to MHC class 1. Now I'll just tell you a rule so that you can remember it very easily for the rest of your lives. This concept of which MHC class presents antigen to which T cell. Just remember a rule of 8. So the product of the CD4 number and the MHC class should be 8. So if it is a T helper cell which has got a CD4 molecule 4 multiplied by what? So that you get 8, you multiply it by 2. So that from that 2 you can actually pick out MHC2. Another rule of 8 is CD8 positive T cell recognizes antigen in context of MHC class 1. So CD8 has got 8 number. You multiply it by 1 to get 8. So CD8 recognizes antigen in context of MHC1. So this is a very simple rule that you can remember. This is a kind of a, you can say a ready reckoner as to which MHC presents antigen to which T cells. So just remember this rule of 8 and this will make your life a bit easier, I hope. Okay. Now, once the CD8 positive T cell recognizes the peptide antigen, which is presented in context with MHC class 1, it gets activated and then it directly kills that same very cell which is presenting that antigen. And this is done by producing chemicals like perforins and granzymes. Perforins, basically, as the name says, creates a hole in the cell membrane through which the granzymes enter, and the granzymes basically activate the programmed cell death pathway which will be taught to you in your pathology classes, which is also called as apoptosis. So that's how CD8 positive T cell actually kills a cell which is presenting an antigen that it can recognize in context of MHC class. Okay. Now, CD8 positive T cell, you will be surprised to know, actually kills its own cell, our own body cell, but that body cell is basically a threat to the rest of the individual. So these are therefore referred to as altered cell cells. If you can see over here, there are two main altered cell cells that it recognizes and tries to remove. That is virally infected cell. You'll learn about viruses a bit later. Viruses multiply within our own cells and use them as a factory. So therefore, if you are able to tag that kind of a cell and then kill it, you prevent the viral replication. The idea is to basically kill your own cell which might pose a threat to the rest of the body. So it's for the larger good we are doing this. Another kind of cell is a tumor cell. A normal cell is turned into a cancerous cell. So it is able to recognize it through the tumor antigens, which are expressed on the surface of the tumor cell, and then kill it. Okay. Now let's talk in terms of SARS-CoV-2. Suppose a person is infected with SARS-CoV-2, it is multiplying in the cells of our body. The antigens of SARS-CoV-2 would be processed, and then some of them would be presented in context of MHC class 1, and CD4 positive T cells having the T cell receptor recognizing those will recognize that particular antigen in context of MHC class 1, and then would kill that our own cell in which the virus is replicating in the hope of preventing the further spread of infection. Hence, CD8 positive T cells or cytotoxic T cells are referred to as having two main properties, the antiviral property and anti-tumor property. This we'll further discuss in the form of a diagram that I'm going to show later. Okay. Now we will shift our attention to B lymphocytes. B lymphocytes are referred to as B lymphocytes because they develop in bursa fabricius. That was an organ in the birds where they were first identified as a B cell. And later on, we realized that in the humans, actually, they are developing in the bone marrow. Unfortunately, that word also starts with B, but the letter B actually stands for bursa fabricius. We discussed this earlier also. The CD markers, which are present, expressed on the surface of a mature B lymphocyte, are CD19, 20, and 21. Unlike T lymphocytes, they develop in the bone marrow as well as mature in the bone marrow itself. There is no thymic education which takes place. So they don't go into the thymus for further maturation. All B lymphocytes, just like T lymphocytes, because they are part of the acquired or adaptive immune response, they display B cell receptors. So what are B cell receptors? B cell receptors are nothing but the antibodies which are expressed on the surface of the B cell. They are bound, that is, they are not released. And these antibodies, the structure of the antibody would have been covered earlier by during other sessions. These antibodies, they are stuck onto the surface of the B cell, by which the B cell recognizes the antigen. Once the antigen binds to the B cell receptor, it gives a signal to the B cell to get activated. And another signal actually also comes from the Th2 cell that we were referring to earlier. And then the B cells, they become activated, they start to multiply, and then transform into plasma cells. Now, what is the difference between a plasma cell and a B cell? Plasma cells, they produce antibodies and then they release it into the circulation. The antibodies are not stuck onto the surface as a BCR. They are actually producing a soluble BCR, if you can put it that way. And these are the antibodies then which bind to the specific antigens which initiated it and try to remove it and then try to resolve the infection. Okay. So this was about the B lymphocytes. So just to show you in the form of a diagram, this is a mature B cell. As the B cell matures, the marker of maturity of a B cell is the surface expression of IgM and IgD. These are the two classes of antibodies which are expressed on the surface of the mature B cell. I'm sure you would have learnt about the classes of antibodies in one of the earlier sessions. And once the antigen binds to the B cell receptors, it gives a signal to the mature B cell to get activated. They start to multiply and form plasma cells, and these plasma cells start to secrete antibodies of various classes. Okay, so this is just how the B cell gets activated. Okay, now let's talk a bit about the lymphoid organs, because you need to know how all of these things play out in the lymphoid organs and tissues. Now, what are lymphoid organs and tissues? As the name suggests, lymphoid tissue and organs, these are the collection of the immune cells, either in the form of an organized organ, that's what is called as an organ, or in the form of a collection, which is referred to as a tissue. Okay, all these structures are collectively referred to as lymphoid systems. All the cells, basically, as I was mentioning earlier, arise from the bone marrow and then they home in into these lymphoid organs. Now, this is a very important concept that you need to understand. There are two types of lymphoid organs and tissues. These are primary and secondary. Now, the primary lymphoid organ is also referred to as a central lymphoid organ, whereas the secondary lymphoid organ is also referred to as a peripheral lymphoid organ. So, whenever I say primary or central lymphoid organ, it means the same. Similarly, secondary and peripheral lymphoid organs, they all happening in the same. Now, what is the difference between a primary and a secondary, or a central and a peripheral lymphoid organ? Let's see. Primary or a central lymphoid organ is the site where the lymphocytes they develop. Okay, they are immature, and then they develop in these organs, and then they go out into the secondary lymphoid organ to then interact with the antigen. There are only two central or primary lymphoid organs that you all have to remember. These are thymus and the bone marrow, and we'll discuss about these two. Starting with the thymus. Thymus, as we've been discussing earlier also, is a site of maturation of the T lymphocytes. It's the first lymphoid organ to develop. It's a tiny organ which is present just behind the sternum, the upper part of the sternum, below the neck. It's a bilobed organ, and it's further divided into two regions: the cortex and the medulla. Now, in this, the T cells are developing. Okay, so this is just to show where the thymus is, and it has got two regions. We do the histology of it. It has got a cortex and a medulla. This is just a diagrammatic representation. Okay, now what happens is that a large number of T cells are developing, and different T cells have got different T cell receptors, and these are randomly generated by genetic recombination. And each of the different T cell receptors will be recognizing a specific antigen. Now, because these T cell receptors, which are expressed on the surface of the developing T lymphocytes, are randomly generated, they may recognize both self antigens, that is part of your own body, as well as non-self antigens, that is the ones which are not part of your own body. Okay? Now, you have to actually now prune it. All the self-reactive T cell receptors have to be removed. So what happens is that the large number of these T lymphocytes which are developing in the thymus, they react with the self antigen. These self antigens are being presented in the thymus, and any T lymphocyte which is developing in the thymus, if it binds with the self antigen, it is automatically deleted from the system, because you don't want any T cell to be there which will be binding and then reacting with a self antigen. Otherwise, you will have a autoimmune reaction. And this whole concept is called as a negative selection. So, what is a negative selection? Negative selection is during the development of T lymphocytes in the thymus, removal of all self-reactive T lymphocytes before they are released into the circulation. So, whatever T lymphocytes are coming into the secondary lymphoid organ would only be the ones which are actually binding in the non-self antigen, and that's what you want to basically attain. And please remember, this is what we refer to as, in simple words, thymic education, wherein all the self-reactive T cell clones are deleted, and the ones which are left automatically are the ones which are going to bind and react and fight against the non-self antigens. Okay, that's what we said in my earlier session. Acquired immune response has got a way to distinguish between self and non-self, and this is how it is attained. Finally, what is left are only the T cells which are going to recognize the non-self antigens and fight against it. Any self-antigen recognizing T cell clone would be deleted. And through this process, more than 95% of the T cells, they are removed. Only the ones which are recognizing the non-self antigens are able to survive and then go into the secondary lymphoid component. I hope this concept is a slightly difficult concept, and so try to make it easy for you to understand. Probably you would have got the point. In the bone marrow, as I said, B lymphocytes arise, and they not only arise, they also mature. And the B cell receptor is the counterpart which recognizes the specific antigen on the surface of the B lymphocyte that we just alluded to. Similarly, they are created randomly on different B lymphocytes, and B lymphocytes also undergo a negative selection. So all the self-reactive B cell clones are deleted, and the ones which are actually going into the secondary lymphoid organs are the ones which will only recognize the non-self antigens. Okay, so negative selection therefore occurs in B cells or T cells. Quick answer, if you're listening to me. So negative selection कहाँ होता है बेटा? B cell में होता है, T cell में होता है कि दोनों में होता है? Good, excellent, good. So it means that you are getting the point. I'm pretty happy that you are able to get this complex concept within your mind. That's how you'll be able to understand the immune response. Otherwise, it becomes very difficult. Okay? So just to show you the bone marrow where the development is happening on the B cells, and also, as I said, it is the place where all the other immune cells also originate. Okay. Okay. So this we've already discussed. It's the stem cell, and these B and T cells are being generated. Any B or T cell, which is recognizing the self antigen over here is deleted, and the ones which are left are the ones which will recognize the non-self. Okay, and if this process fails, this fails sometimes. It is not a foolproof process. In such a situation, person develops autoimmune disease. So this is one of the reasons for development of the autoimmune disease: non-removal of self-reactive B cell T cell flow. Now we come to secondary or peripheral lymphoid organ. Once the T and B cells have learned, they've undergone their education and clonal deletion. Clonal deletion is the other word that we use for negative selection. These T and B cells go into the secondary or the peripheral lymphoid organs. Now they are mature and they are ready to fight. Okay. What are the peripheral or the secondary lymphoid organs? These are lymph nodes, spleen. These are the two organs, and then there is a tissue which is referred to as mucosa-associated lymphoid tissue. Mucosa-associated lymphoid tissue. Okay, mucosa-associated lymphoid tissue. Now we'll try to understand each of these organs and tissues as we move along. Starting with the lymph node. Lymph nodes are numerous. They are distributed all across our body, and they are bean-like structures, tiny bean-like structures. They are divided into, each of them are divided into cortex, paracortex, and insula. Now these are the questions that you sometimes get. What is a B cell in a T cell area? Cortex is basically the B cell area, and the paracortex, which is surrounding the cortex, is mainly a T cell area. The idea is basic of the lymph node is basically the lymphatic vessels. As you can see, these are the tiny lymph nodes, and these are the lymphatic vessels which are entering, and then these are called as afferent, and then the ones which are leaving are called as efferent. So the tissue fluid which is being generated is passing through these lymphatic vessels, and if there is a threat in any of these tissue fluid in the form of an infection or a offending pathogen, they are trapped in the lymph node, and T and B cells then do the necessary work to remove the pathogen, okay, and fight against it. So just to show this bean-like structure, you've got this paracortex over here. This is a paracortex, this area, which is a T cell area. Okay, and then after that we've got the cortical area. This is the cortex, which is the cortical area, which is the B cell area. This is the histology of it, and all of these I'm sure in your anatomy classes as well as in your pathology classes will be discussed in more detail. So I'm just going to give you an overall picture, not the details at this point with regard to the histology of it. Spleen is another peripheral or the secondary lymphoid organ. It is the largest lymphoid organ, and be always present on the left upper quadrant of the abdomen. It has got the white and the red pulp. White pulp is the one that we are interested in as far as the immune system is concerned. It is not supplied by any lymphatic vessels. It has got its own splenic artery and vein that we all know. Okay. The white pulp, you've got a splenic arteriole, and around the arteriole you have a periarteriolar sheath, which is the area which is rich in T cells. Around it is the germinal center, which is rich in B lymphocytes. Okay. The main purpose of spleen is basically to fight against blood-borne pathogens, as opposed to lymph nodes. The main purpose of lymph nodes is to be ready to fight against any pathogen which is entering through the tissue, and then it travels to the lymphatic vessels and it is trapped within the lymph nodes, and then the specific T and B cells fight that infection. Whereas spleen does the same job, but it protects our bloodstream. Okay, so just to show you the histology of it, this is the central arteriole which is coming out of the splenic artery, and around it you've got the periarteriolar sheath which is rich in the T cells, and then we've got the germinal centers, which is also referred to as a follicle, which contains the T cells. Finally, we come to the tissue and not an organ, that is MALT. MALT, we understood earlier, stands for mucosa-associated lymphoid tissue. This is not organized in the form of an organ, so therefore it is referred to as a tissue. So this is present in various mucosa like intestinal tract in the form of tonsils, Peyer's patches, genitourinary tract, tracheobronchial tree, Okay, and as is obvious, based on its anatomical location, it helps in fighting against infections which are occurring through the mucosal surfaces, that is, through the respiratory tract, genitourinary tract, gastrointestinal tract. So therefore, we've got three peripheral or secondary lymphoid organs which protect us from three sides. So lymph nodes are protecting us against any infection which is invading through the tissues. Spleen is protecting us from any infection which is spilling into the blood, and mucus-associated lymphoid tissues protecting us from any infection which is coming through the mucous membranes. Play an extremely important role. We actually survive and are healthy most of the times thanks to these lymphoid organs and tissues. So just to show you the mucosa-associated lymphoid tissue, this is the mucosa, the mucosal membrane, and inside it is the collection of these immune cells. Okay. Now, I'll give you we'll just digress a bit. Now, I will talk about major histocompatibility complex because this is what we are going to discuss subsequently. The major histocompatibility complex is a protein basically, which is coded by the genes which are located on short arm of chromosome 6. So this is a very common question that you get in your exams. The protein which is expressed is called as a human leukocytic antigen, or HLA. So when I say HLA, human leukocytic antigen, it is a protein part which is expressed by a group of genes which is referred to as major histocompatibility complex. They are very important molecules because they are very unique in every individual. So they play a very important role in allograft rejection. This will be taught to you in your pathology classes or maybe in microbiology later on. And also they play a very important role in immune response in the form of antigen presentation that we are going to come to. There are three classes, 1, 2 and 3. The role in allograft rejection in antigen presentation is only of class 1 and class 2. Class 3 has got no role. Okay? So class 1 is further subdivided. I'll just repeat. Somebody said, Adiba. I'll just repeat. HLA beta stands for human leukocytic antigen. So it is a protein which is expressed on the surface of the cell. Human leukocytic antigen. Okay. Now this protein is coded by a set of genes. Those set of genes are referred to as major histocompatibility complex, or MHC. So MHC is a collection of genes. Whereas the same genes, if they are expressed as proteins, these are then referred to as HLA. Is that clear, Adiba? Good. So these genes, you divide them into three classes, 1, 2 and 3. 1 and 2 are the ones which are involved in our immune response. 3 is not involved. Okay. Now, let's see the classes. Class 1, Class 2, Class 3. Class 1 has got 3 loci, and we call them as loci A, B and C. 3 loci, A, B and C. A, B and C. Class 2 have got again 3 loci which are called as DP, DQ and DR. Okay, the moment I say HLA, A, B or C, automatically one should understand that I'm going to talk about class 1. Similarly, the moment I say HLA, DP, DQ, DR, it means I'm talking about class 2. Class 3 has got nothing to do with the antigen presentation. However, it codes for certain important genes which play important role in the immune response, but not in antigen presentation. They code for complement component 2, complement component 4, factor B of the alternate pathway. Remember complement system that we talked about, and then certain cytokines like TNF alpha and beta. Now, to further complicate the matters, each locus has got multiple alleles. They are alternate forms of the genes. So, class 1, A, B, C loci. A has got multiple alleles, B has got multiple alleles, C has got multiple alleles. Similarly, class 2, DP, DQ, DR, multiple alleles of each one. Okay. So, these are the multiple forms of the antigen. So, A has got about 24 alleles, B has got 50 alleles, so on and so forth. Okay. As I said earlier, class 1 and class 2 bind to the peptide antigens and present it to the T cell. Class 1 presents to cytotoxic T cell, whereas class 2 presents to helper T cells. Let's see the structure of class 1 and class 2 now. Class 1 MHC molecule has got 2 chains, alpha chain, and there is something called as a beta 2 microglobulins. Let's see. I'll show you the structure over here. So this is a MHC molecule structure. This is an alpha chain, and this is a beta 2 microglobulin. Alpha chain is further divided into 3 domains: alpha 1, alpha 2, alpha 3. Okay. Now this is the cleft which is formed between alpha 1 and alpha 2 is the one which actually binds to the peptide fragment and presents to cytotoxic T cell. Okay, so I'll just go back now. So alpha chain and beta 2 microglobulin, and it's the domain between alpha 1 and alpha 2 which presents the peptide fragment to cytotoxic T cell. Now this is a very common question that you get in your exams: which cells express MHC class 1? They are expressed by all the cells except RBC. So all cells of our body express the MHC class 1 except the RBC. And what are the peptide antigens they present? They present the endogenous peptide. Now what do you mean by endogenous? Any peptide which is produced within our own cell is called as an endogenous. Now there are only two endogenous peptides that you have to remember. So if your own cellular machinery is being used to synthesize a peptide through translation, this is called as endogenous. There are only two endogenous peptides you will realize. One is the tumor antigen. So if a normal cell is turned into a tumor cell, it synthesizes its own unique proteins that can be then presented. And then the other one is a virally infected cell. So as you know about viruses, when a virus infects the cell, it uses the cellular machinery for its own synthesis of its own proteins. So there are two main endogenous peptide antigens that you all need to remember. These are the tumor antigens and the viral antigen. And then these are presented to cytotoxic T cells which recognize it, and then they are able to fight it and kill it. Hence, we mentioned earlier also, cytotoxic T lymphocytes therefore have got two main properties: antiviral property and anti-tumor property. So I hope you are able to make the connect that is desirable to make. Now we come to MHC class 2. MHC class 2 has got two chains again, alpha chain and beta chain. Both the chains, alpha chain and beta chain, have got two domains each. You've got two domains each, alpha 1, alpha 2, beta 1, beta 2. And it is the cleft which is formed between the alpha 1 and beta 1 is the one which presents the peptide antigen. Okay, so let's go back. So we've got alpha chain, beta chain. And it's the peptide antigen binding group is formed between the alpha 1 domain and the beta 1 domain. So it's incorrect, just correct it. It is alpha 1 and beta 1, not beta 2. Okay. Now, where do the MHC class 2 molecules are expressed? Now, it's an extremely important question. All cells do not express MHC class 2. Please remember, is a rule. Only specialized cells express MHC class 2. These are called as antigen presenting cells. Antigen presenting cells. So only specialized cells express MHC class 2 as opposed to MHC1. MHC1 is expressed by all cells except the RBCs. Whereas MHC2 is on the surface of only specialized cells, that is antigen presenting cells. The main antigen presenting cells that you need to remember, again this is a question that you get in your exams. Dendritic cells are the most efficient antigen presenting cells. And then we have monocytes, macrophages, and also the lymphocytes also function as antigen presenting. Now, these antigen presenting cells, they eat up the pathogen which is causing the infection, break down the protein of that pathogen, and then present it along with MHC class 2. Hence, they present exogenous peptides. So, another difference, MHC class 1 presents endogenous peptides which are synthesized within the cell by using the cell's own machinery, hence tumor antigens and viral antigens. Whereas antigen presenting cells, they phagocytose the pathogen in question, break down the protein of that pathogen into small peptides, and then present it in context of MHC class 2 to helper T cell. Okay, so I hope you are able to get the finer nuances with regard to MHC class 1 and class 2 and antigen presentation. So this diagram we've already covered. Okay, so just to show you this pathway, endogenous pathway and exogenous pathway. The endogenous pathway is for MHC1. The peptide is synthesized within the cell. It is presented in context of MHC class 1 onto the surface. Whereas exogenous pathway, you phagocytose the pathogen. Its proteins are broken down in the phagolysosome, and some of the peptide fragments are loaded onto MHC class 2 and then presented on the surface of the cell. So just I hope you would have got this point. Now this is the most important slide that I'm going to show how actually the immune response develops. Let's see how actually the immune response develops. Now, first we'll talk about the helper T cell activation and its effector functions, and then I'm going to talk about cytotoxic T cell, and finally I'm going to talk about the B cells. Now, the exogenous antigen is eaten by the antigen presenting cell, broken down by the phagolysosome into tiny fragments, it is presented in context of MHC class 2 to the T cell receptor of the T helper cell. Okay. Once the T helper cell recognizes this antigen, it gets activated. This T helper cell gets activated in one of the two forms. These two forms are Th1 cell and Th2 cell. These are the two functional subsets. And this depends upon the local presence of certain cytokines. And these are then referred to as Th1 cytokines and Th2 cytokines. The important Th1 cytokines are IL-2, IL-12 and gamma interferon, while important Th2 cytokines are IL-4, 10 and 13. If the T helper cell gets activated into Th1 cell, this Th1 cell then in turn activates macrophages. These macrophages, though they are able to phagocytose the pathogen, they become very activated upon the help provided by Th1 cell, and these are then referred to as activated macrophages. And then they are able to kill the pathogen. The main action of these activated macrophages is against the intracellular pathogens. The main action, like in Microbacterium tuberculosis, like in Microbacterium leprae, they mainly act against it. On the other hand, if a T helper cell gets activated into a Th2 subset, because of the type of the antigen, the type of pathogen, and the local milieu of cytokines, this Th2 cell in turn activates B lymphocytes. And this B lymphocyte, in turn, once it gets activated, converts into plasma cells and releases antibodies. And as I said, antibodies can only damage the extracellular pathogen because antibodies cannot enter inside the cell. So basic action against these antibodies is basically against the extracellular pathogen. So that's the central theme that you have to understand. If you understand this diagram, a lot of your questions would be basically answered. Now, because in Th1 response, cells are involved in damaging the intracellular pathogens like macrophages and Th1 cell, this is called as a cell-mediated immunity. Whereas in Th2 cell, in turn activates B lymphocytes and then antibodies are formed which activate and then which are released and then fight against extracellular pathogen, and they are there dissolved in the circulation. This is referred to as a humoral immune response. So Th1 cells are main mediators of the cell-mediated immunity, whereas Th2 cells are main mediators of the humoral immunity. So ultimately, cell-mediated immunity and humoral immunity is decided by which cell? Quick answer. It is decided by which cell? Can you give an answer, beta? Quick answer. Very good. It is the helper T cell. Very good. It is the helper T cell which decides whether you will develop a cell-mediated immune response or the humoral immune response. Hence, your helper T cell is the most important cell of your adaptive immune system. If you have deficiency of helper T cell, you will become immunosuppressed, and that unfortunately happens in HIV. I'm going to talk about HIV tomorrow. HIV virus actually damages the most important cell of your adaptive immune system, that is the helper T cell, and that's why you become immunosuppressed. Okay, good. Now we come to cytotoxic T cell function. Let's integrate the knowledge that we have developed till now. So cytotoxic T cell recognizes the antigen. This is the cytotoxic T cell which recognizes the antigen in context of MHC class 1. MHC class 1 is expressed, as I said earlier, on our own cells which have become defective, either virally infected or tumor. So the peptide antigen is created inside the cell. It is loaded onto MHC class 1, recognized by T cell receptor of a cytotoxic T cell. Once it gets activated, it then produces porins and granzymes and then damages the cell. And because it directly kills the cell, the altered cell, virally infected cell or a tumor cell, this is called as a cytotoxic T cell. Cytotoxic. And the main action of cytotoxic T cell, I'm again repeating, is against what? Quick answers, beta. The main action, because the type of antigen it recognizes, the main action of cytotoxic T cell is against what? Quick answer. Batao beta. Because it recognizes the endogenous antigens, which are of only two types, tumor antigens and viral antigens. Hence, the main action of cytotoxic T cell is only against tumor cells and virally infected cells. Excellent. Kirti, Anshub, you're giving wonderful answers. Pretty happy. Okay. This we've covered. Antibody structure you've already covered, so I'm not going to talk about the antibodies. I'll mention over here how do antibodies bring about their effector function. The how the immune response develops with regard to the humoral or the antibody response. So the antigen presenting cell presents the exogenous antigen to the T cell present to the T cell receptor. T cell gets activated into a Th2 cell. Th2 cell then activates the B lymphocyte. B lymphocyte, once it gets activated, multiplies and converts into plasma cells, and antibodies are secreted by the plasma cells. Now, what do these secreted antibodies do? Let's see. What do the secreted antibodies do? What do the secreted antibodies do? Antibodies, they bring about an immune response by various mechanisms. One of them is neutralization. These are the effector functions of the antibody. Neutralization. Neutralization is the antibody directly binds to the pathogen or the toxin and does not allow its entry. Now, we are talking a lot about neutralizing antibodies with regard to SARS-CoV-2. हर कोई बोल रहा है neutralizing antibody बनी नहीं बनी. So this is what we mean by the word neutralizing antibody. All antibodies are not neutralizing. Only the ones which are able to bind the pathogen and prevent its entry into the cell are referred to as neutralization or neutralizing antibody. Okay, and then these antibodies which are bound to the pathogen or toxin will then removed from the system by phagocytosis. Now, what are neutralizing antibody? Let's see in the form of a diagram. This is a microbe. Let's say a virus. This virus binds to the surface of the host cell and then multiplies within it and damages it. Now, how can I prevent an infection caused by this kind of a virus? You develop antibodies against it. These antibodies bind to the exterior surface of the virus, let's say. Because they bind to the exterior surface of the virus, it does not allow that particular virus to interact with the surface of the cell and then enter. And such antibodies are therefore, as I was mentioning earlier, are referred to as neutralizing antibodies. Neutralizing antibodies. The same thing can also happen with the toxin. Suppose there are some pathogens like Corynebacterium diphtheria that you're going to learn later on in bacteriology, which produce a toxin which actually damages. So similar way, if you form an antibody against the toxin and does not allow the toxin to enter the cell, you effectively prevent the action of that particular toxin. And such antibodies are referred to as antitoxins. You'll learn about these later on. The second effective function of an immunoglobulin is another name for antibodies, opsonization. This we learned last time also, if you remember. Opsonization is, we talked in context with complement, which is the most important complement component which is opsonizing. We learned about that, C3b, that we learned in our last session. Similarly, there are certain antibodies also which are opsonizing. Now, the most important antibodies which are opsonizing are IgG class of antibody. So what does it do? The IgG antibody binds to the antigen. The other part of the antibody has got a receptor on the surface of the phagocyte. So the phagocyte has got a receptor for the Fc part of the IgG. Remember I talked about Fc part and Fab part. Fab part is the one which binds with the antigen. Now the Fc part undergoes a conformational change when the Fab part binds with the antigen. And only when this conformational change happens, it is able to bind to the receptor which is normally present on the phagocyte. So that's how the receptor binds to the Fc part and then in turn the antibody is bound to the antigen. This is then internalized by phagocytosis and then it is the pathogen is killed. And this is referred to as opsonization. If you remember, this can also be done by the complement component. If the pathogen has got C3p deposited on it, phagocytes also have got a C3 receptor through which it is able to bind to C3 which is deposited on the antigen and then it is ingested and then it is killed. So there are two ways of opsonization. One is an antibody, the other one is a complement component C3b. It was attentive in the class of complement taken last time. Okay. So this is opsonization. There is complement fixation that we discussed in detail last time, so it should be easy for you. So antigen antibody complex is formed. This activates the classical pathway of complement system. C3b deposition occurs and then MAC is formed. C3b deposition leads to opsonization and MAC deposition in the surface of the virus or the bacterium, particularly a gram-negative bacterium, creates a hole. The leakage of the cytoplasm occurs in the cell dies. So complement-mediated damage also occurs. So what I'm trying to say is that the antibody either can directly neutralize the antigen or the pathogen or the toxin, or after binding, it can recruit other mechanisms like opsonization or complement and then get rid of the pathogen. Okay. This we've covered. And finally, is antibody mediated or antibody dependent cell mediated cytotoxicity. Short note आता है बेटा आपका. That's another mechanism, antibody-dependent cell-mediated cytotoxicity. What is antibody-dependent cell-mediated cytotoxicity? The antigen, if it is in the form of a cell, is bound by the antibody. The antibody Fc part has got receptors by certain cells. These cells are phagocytic cells. These cells recognize the antigen and then kill it. So this is called as antibody-dependent cell cytotoxicity, or ADCC. This is also brought about by what we refer to as NK cell. Okay? So NK cell also does this job by virtue of having a receptor for the Fc part. I've got a question from Akshara. Sir, how can Ph1 cells act against intracellular pathogens as helper T cells are activated by MHC2 molecules which are exogenous? Now, very good question that you've asked. The MHC2 basically, let's say talk about Microbacterium tuberculosis. Okay, Microbacterium tuberculosis is multiplying inside the cell. Okay, this Microbacterium tuberculosis, if it is multiplying inside the cell, will be phagocytosed, let's say, by a macrophage. Now, this Microbacterium tuberculosis can also be ingested by a macrophage. The Microbacterium tuberculosis which is ingested by the macrophage, the proteins of that particular pathogen would be broken down into tiny, tiny fragments. One of these peptide fragments of Microbacterium tuberculosis would be presented in context of MHC class 2. This MHC class 2 presentation to a TH cell in the presence of right milieu of cytokines will convert it into a Th1 cell. This Th1 cell will in turn activate the macrophage, and these macrophages, once activated, are better capable of killing the pathogen. So they'll surround the intracellular pathogen which is there inside a cell, and these macrophages would indiscriminately eat whatever is there in front of them and kill it. So now, Akshara, tell me, how can the macrophage kill? Macrophage is part of the innate immune response or acquired immune response? Quick answer. हमने पढ़ा था macrophage innate का part है, acquired का. Very good. It does not have eyes. It cannot see the pathogen. So what is giving that direction of killing and power of killing? It is the Th1 cell. Th1 cell has got eyes because it has got receptor by which it can recognize the pathogen. Are you getting the point? So this is the classical example where the adaptive immune response, that is the acquired immune response, and the innate immune response work together to get rid of the effect. I hope I've been able to clear. It's difficult for me to actually explain online. Had it been a face-to-face class, probably I would have done more justice to it. Very good question, Kshitij. IgM, how does an IgM antibody act opsonize? Now, please remember, IgM itself does not have opsonizing activity. IgM opsonizing activities indirectly occurs through complement activation. So IgM is basically a pentamer. So it activates, once it binds to the specific antigen, it activates the classical pathway of the complement system, and it causes the deposition of C3b near the pathogen. And this is the one which actually brings about the opsonization. Directly, IgM does not have very good opsonizing activity, but indirectly, through the activation of the classical pathway and deposition of C3b, it is able to have an indirect opsonization activity. Direct opsonization activity is that of IgG and to some extent of IgA, but not of IgM. IgM indirectly basically brings about. I hope Kshitij have answered your question. So let's move forward. So these were the effector mechanisms of the humoral immune response. So with this we come to the end. I'll give you some questions as is customary in my sessions, just to see whether you've been able to understand. So what is the CD marker which is used for the identification of T lymphocytes? CD4, CD8, CD16 and CD3. So a pan T lymphocytic marker, we all know, is what? Quick answer is CD3. Good. Most of you have answered the question right. So any cell which has got CD3 which is present on the surface is referred to as a T lymphocyte. Okay. If a cell has got CD3 and CD4 present on the surface, then it will be called as a T helper cell. If a cell has got CD3 and CD8 on its surface, we then will call it as a cytotoxic T cell. Finally, CD16 and 56 we discussed are the markers for identification of NK cells. CD16 and 56. So please remember, these are very common questions that you get in exams, right? Pooja, you're absolutely right. Okay, now question number 2. CD8 positive T cells are restricted to which self-MHC molecule? MHC1, MHC2, MHC all. Remember the rule of 8 that I talked about? This will make your life easier. CD8 का 8 उठाओ. How much do you need to multiply with to get 8? 1. So therefore, by this rule, if you remember it, CD8 T cells are restricted to MHC class 1. Okay. Similarly, rule of 8 for CD4. CD4 into 2, 4 into 2, MHC2. So CD4 T alpha cells restricted to MHC class 2. So this is very simple way to. Number 3, negative selection occurs in which cell? T cells, B cells, both, none. Quick answers, beta. Remember? Excellent. Very good. So negative selection occurs during the maturity of T cells in thymus and B cells in bone marrow. They randomly generate their TCRs and BCRs. Because they are randomly generated by genetic recombination, they will recognize both self and non-self antigens. So therefore, they need to have an editing mechanism wherein you remove all the all the self-reactive clones. Therefore, whatever T and B cells are maturing and entering into the secondary lymphoid organ are the ones which only recognize the non-self. Breakdown of this mechanism, as I said, is life-threatening and can lead to autoimmune disease. Negative selection is also referred to as clonal deletion. So please remember there are 2 words which are used for it: clonal deletion or negative selection. Good. Excellent. Question number 4. All are peripheral lymphoid organs except lymph nodes, spleen, thymus, malt. So all of the following are peripheral or the secondary lymphoid organs except, very good, thymus. So there are only 2 central or the primary lymphoid organs, thymus and bone marrow. Rest all the others, spleen, lymph node, malt, are they all secondary or peripheral lymphoid organs. Please remember, आपको ये बेटा सवाल आता है, स्प्लीन को कभी भी सेंट्रल लिम्फॉइड ऑर्गन मत बोलना. पता क्या हो जाता है, आपका पता नहीं कुछ हो जाता है एग्जाम के टाइम पे, आप सोचते यार स्प्लीन है, पेट के अंदर है, सेंट्रली लोकेटेड है, तो उसको सेंट्रल लिम्फॉइड ऑर्गन कर देंगे. नो, दैट्स अ वेरी कॉमन मिस्टेक दैट यू डू later on. I know these are the kind of mistakes that you do in your entrance exams. So please remember, it depends upon the maturation. So if the naive T and B cell is getting matured in a lymphoid organ, that's a primary or a central. So in case of T cell, it is thymus. In B cell, it is bone marrow. Where the mature cells reach and they wait for the antigens to react and fight, this is then referred to as a secondary or a peripheral lymphoid organ, which includes lymph nodes, spleen, and malt. Good. We come to the final question now. The main action of Ph1 cell is against tumor cells, intracellular pathogens, extracellular pathogens, all. So main action of Ph1 cell is against tumor cells, extracellular pathogens, intracellular pathogens, extracellular pathogens, all. Ph1 cell का बेटा main action क्या है बताओ? Ph1 cell का बताओ बेटा? Ph1 cell does what? Ph1 cell in turn activates the macrophages. These macrophages, once they get activated, they get extra power of killing. They surround themselves with the infected cells, that is cells infected with Microbacterium Tuberculosis or Microbacterium Lymphoids, just to take an example, and kill it. So the main action of Th1 cells is against intracellular pathogens. Intracellular pathogens. Okay. Whereas what is the main action of Th2 cell, beta? What does Th2 cell do? Th2 cell activates B lymphocytes. B lymphocytes, once they get activated, convert into plasma cells. These plasma cells secrete antibodies. The antibodies, please remember, as a rule, can only bind with an extracellular pathogen because antibodies cannot enter your own cells. Okay, so therefore the main action of Th2 cell through B cells and antibodies is against an extracellular pathogen. हमारी एंटीबॉडी बेटा हमारे अपने सेल के अंदर नहीं घुस सकती, ठीक है? तो हम कभी भी एंटीबॉडी इंट्रासेलुलर पैथोजन को नहीं मार सकते. Is it clear the concept? Okay. So with this, I think sincerely hope that this session was a difficult one, but I hope you've been able to build up your concepts of immunology, because if you develop these concepts at this stage, the rest of the other things which use these concepts will become very easy for you to really understand. So thank you very much for listening. If you have any queries, you can post in the queries either through my email address, okay, or you can, difficult times you can't meet, but anyway, through any of the channels you can contact me and I'll try to answer your questions. Maybe later on when we do a face-to-face class, so we'll further discuss issues and try to resolve those issues. This is inside my book. If you want more concepts, you can go through it. It's your wish. Thank you very much. It was wonderful interacting with all of you. Have a great day.